students, coming to this class, we had discussed about nomenclature and the classification of enzymes. In the last class, we discussed about the characteristic features of the enzymes and composition of the enzymes and the velocity graph. We have discussed about these 3 points in the last class. Coming to this class, nomenclature and classification of enzymes. See, naming to enzyme, the process is called as nomenclature. We know, we already discussed in botany, naming to any organism, that is the nomenclature process. Here also, naming to the enzymes. So, that process is called as nomenclature. And next one is classification of enzymes. See, here classification of enzymes means based on type of reaction. See, a group of enzymes are there. Some enzymes performs one kind of reaction. Another group of enzymes performs another kind of reactions. So, what I am telling here, based on type of reactions, the enzymes are classified. The enzymes are classified. So, here nomenclature and classification of enzymes we are discussing, and based on the type of reaction, the enzymes are classified into 6 types. The enzymes are classified into 6 types. See, simply this is also called OTHLIL. OTHLIL. So this is about the shortcut identification for the different types of the enzymes, totally based on nomenclature and classification. See here, nomenclature depends on the reaction and classification also depends on reactions. So based on type of reactions, the enzymes nomenclature and classification done. Totally we have 6 types of enzymes. So in that 6 type of enzymes, the shortcut name is here OTHLIL, right? So O indicates oxidoreductases and T indicates here transferases, H indicates here hydrolases, L indicates here liases, I indicates here isomerases, and last one L indicates here ligases. Totally I said 6 types of enzymes are there. So total enzymes can be classified into 6 types based on the reaction, type of the reaction. So the very first one is oxidoreductases. So the name indicates, say a group of enzymes performs oxidation reactions or reduction reactions. We know oxidation is nothing but, for suppose in a reaction, if we add the oxygen, that is oxidation. If we remove the hydrogen, that is also oxidation. See, in the reduction reaction, addition of hydrogen atoms, removal of oxygen atom, so that is the reduction reaction. So, a group of enzymes which performs oxidation and reduction reactions. A group of enzymes which performs oxidation and reduction reactions. These are, okay, another way is also there, LEO. Losing of electrons, that is called as oxidation reaction. Losing of electrons, that is called as oxidation. Gaining of electrons, that is called as reduction. So LEO, losing of electrons, that is called as oxidation. Gaining of electrons is called as reduction. So based on the type of the reactions here, 2 types are there. Within one enzyme performs here oxidation and reduction reactions. So this kind of enzymes are called as oxidoreductases. The best example is cytochrome oxidase enzyme. So that is about the first type of enzymes. And next one, transferases. Transferases means here a specific substrate is there. 2 substrates are there. From one substrate to another substrate, specific groups are transferred. We know about amino acids. One amino acid is there, one organic acid is there. From this amino acid, the amino group is transferred from this amino acid to another organic acid. So here transfers the amino group. Best example, for suppose this is one substrate, and this is another substrate. This is one substrate and this is another substrate. During reaction process, see, one substrate contain group. Okay, that is transferred to the another substrate. another substrate. So this kind of transfers, okay, done by the transferase enzymes. This kind of transfers done by the transferase enzymes. So transferases, these are the enzymes which are transfers the specific group from one substrate to another substrate. So best example here, transaminase enzyme. It transfers the amino group. Hexokinase enzyme. So it transfers the phosphate group. It transfers the phosphate group. Kinase here always related to that phosphate molecules. Right? And next one, hydrolases. Hydrolysis. Lyase means breakdown. Hydro indicates here water. So presence of water molecules. Presence of water molecules. The enzymes. Okay. Presence of water molecules. The enzymes involving cleavage reactions. Cleavage reactions are nothing but the substrate molecules break down into products. One substrate molecule, okay, divided or break down into products. Presence of water molecules. Presence of water molecules. The substrates break down into two product molecules or more than two products. More than two products. So that kind of enzymes are called as hydrolysis. Hydro indicates here water. Okay, lysis. This is indicates here breakdown process. So presence of water molecule, substrate molecules are breaking down. So breaking down of substrate, presence of water molecule. Proteases, lipases, the best example, whatever that digestive tract contain different types of protease enzymes are there, which are involved in cleavage of proteins. Pepsin is there, trypsin is there, chymotrypsin is there, right? Different types of proteases enzymes are there. That proteases and lipases, a group of enzymes which are involved in digestion of lipids. A group of enzymes which are involved in digestion of lipids. These are called as lipases. So here proteases and lipases which are related to the hydrolysis, right? And next one, lysis. Breakdown of substrate without requirement of water molecule. See, these enzymes are involved in breakdown process, and lysis also involved in breakdown process. But here breakdown process or the cleavage process occurs presence of water. But lysis without any requirement of water molecule, the cleavage occurs. The enzymes, a group of enzymes which are involved in cleavage of substrate molecules, absence of water molecules, or without any requirement of water molecules. These enzymes are called as lysis. The best example is the aldolase enzyme. Aldolase enzyme, this is one of the important enzyme for the glycolysis process. Whatever that glucose molecules we are eating, okay, the glucose molecule, the first cycle, the first one is the glycolysis process. Okay, within the glycolysis process, aldolase enzyme involved in the splitting of fructose 1,6 bisphosphate molecules. Fructose 1,6 bisphosphate molecules. Later we'll discuss about that aldolase reactions. So here the example for the lyase is aldolase, right? And next one is isomerases. Here isomerases are nothing but a group of enzymes which are involved in rearrangement of intramolecular structures of substrates. Intramolecular structures of substrates. See, very simple to remember. Remember, all of you, the reason glucose molecular formula is C6H12O6. C6H12O6. This is the glucose. This is the glucose. And coming to the next one is fructose. Here fructose also C6H12O6. Here this one is the fructose. There is no differentiation. There is no differentiation in the molecular formula. There is no differentiation in the molecular formula. But arrangement of atoms are different. Arrangement of atoms are different. C6H12O6. Generally we know glucose. Here C6H12O6. There is also fructose. So there is no change in molecular formula, but orientation of function groups or arrangement of hydrogen atoms, arrangement of oxygen atoms, these are different. These are different. So what I am telling here, isomers, okay, which are having same molecular formula, but the arrangement is different. So rearrangement of intramolecular structures of substrate. The best example is exose isomerase enzyme. Exose isomerase enzyme. See, by the action of exose isomerase enzyme, the glucose is converted into fructose. The glucose is converted into fructose. See, the glucose, this is an aldose sugar. Aldose sugar is converted into keto sugar. Aldose sugar is converted into keto sugar. Not only this one, from glucose, mannose production takes place. From glucose, galactose production takes place. See, by the changing the arrangement of atoms. So that is about isomers which are having same molecular formula, but the arrangement of atoms are different. Right? So that is about that see, in chemistry there is an isomerization is there. See, a group of enzymes which are involved in the isomerization reactions. See, one substrate is converted into the another product. Again, that product, converted into the same substrate molecule. See what I am telling here, which are having same molecular formula, but the arrangement is different. Right? Last one, ligases. See, which are involved in joining reactions. A group of enzymes which are involved in joining reactions. One of the best example, NSL nucleic acids are there. Nucleic acids which are made up of with nucleotides. How the nucleotides are binding? See, if I ask the question, one nucleotide is binded with another nucleotide by the phosphodiester bond. How the phosphodiester bond formation takes place? A group of enzymes which are involved in the formation of phosphodiester bond. So here also, formation of covalent bond between two substrates. There is two substrates are there, which are one substrate is there, another substrate is there. Between these two substrate molecules, there is a covalent linkage formation takes place. See, by the formation of covalent linkage, finally by these enzymes, large structure molecules are formed. The best example, DNA synthesis, RNA synthesis, all these are done by that. See here ligase. Mostly here DNA synthesis process. Within the DNA synthesis process, there is a lagging strand is there. Within leading strand and within lagging strand. See what I am telling here. During DNA synthesis process, this is one of the strand. One strand is during the DNA synthesis process, one strand is continuously synthesized. Another strand discontinuously synthesized. So joining of these two strands done by the DNA ligase enzyme. So whatever that we already discussed, whatever that Okazaki fragments are present, the Okazaki fragments joining process done by the DNA ligase enzyme. So this is about that nomenclature and classification of enzymes. I said here othilin. See, there is an enzyme commission number is also there. Enzyme commission number to that enzymes. See, oxidoreductases. See, the enzyme commission number started with 1. And transferases, the enzyme commission number started with 2. The hydrolases, the enzyme commission number started with 3. So ligases started with 4. Isomerases, the enzyme commission number started with 5. And again ligases, here enzyme commission number is the 6. So here enzyme commission numbers also there. See, that's why given here othilin, here O1, T2, right, H3, L is indicates here ligases, right, 4 isomerases, 5 last one ligases, 6, right. So this is about that enzyme commission number. So enzyme commission numbers for the enzyme. So finally this is about that nomenclature and classification of enzymes and enzyme commission numbers. Two different groups of enzymes. Right. Coming to the next topic, factors affecting enzyme activity. See, different types of factors are there which affects the enzyme activity. Enzyme activity is nothing but we already discussed carbonic anhydrase enzyme, presence of enzyme, right? Within short time the reactions are completed. So enzymes are active, then the reaction will be completed within the short time. Enzymes are inactive, the reaction for the completion of reaction maximum time is taken. So the enzyme activity is also very important. So different types of factors are affecting that enzyme activity. Very first one is that pH. All we know the H plus ion concentration. that is nothing but negative logarithm of H plus ion concentration, that is called as pH. See, different kinds of cells are there, and different kinds of cells and different types of pH are present. One of the best example, within human body, the blood, there is a neutral pH is there, right? And within that digestive tract, there is an acidic pH is there, and there is an tears are there, okay? There is a basic pH is there. So within human body, different types of pH are maintained. In all these conditions, okay, either gastrointestinal tract, exactly within that stomach, there is an gastric juice is there. Always the gastric juice pH is acidic. A group of enzymes are present. That enzymes are only active at acidic pH. And a group of enzymes are active in the neutral pH. Mostly our cell cytoplasm contains enzymes are active at the neutral pH. So one of the TS, okay, there is a lacrimal gland secretes the TS. Within the TS, there is a lysozyme enzyme is present. That lysozyme enzyme active at basic pH. So different types of enzymes and different pH are required. So at particular pH, the enzymes are active. A specific enzyme active at specific pH. So most of the enzymes, or majority of the enzymes, active at the neutral pH. Majority of the enzymes are active at the neutral pH. So here my question is, why enzymes active at different pH? A specific enzyme active at specific pH. So then most of the students have the doubt here. So why enzymes are active, different enzymes are active at different pH? All these are made up of with the amino acids. All these are made up of with the amino acids, right? No doubt about it. Every enzyme is made up of with the amino acids except the ribozyme enzyme, right? See here, pH. Okay, first of all, there is an enzyme is there. If the enzyme contains amino acids are active at acidic pH, the enzyme active at acidic pH. And one protein or one enzyme contain amino acids. Amino acids are active at basic pH, the enzyme active at the basic pH. If the enzyme contain amino acids active at neutral pH, the enzyme active at the neutral pH based on the amino acid and pH. Based on amino acid and pH. Favorable pH is there. Amino acids are active. Okay, then within that active site, there is a structural conformation changes that takes place. So within short time, the reactions are completed. Within short time, the reactions are completed. For suppose this is the enzyme. The enzyme requires acidic pH. Then amino acids are more flexible. If amino acids are more flexible, substrate is easily binded. There is a stable substrate enzyme complex formation. Finally, product formation takes place. The same enzyme, generally the enzyme requires acidic pH. But if neutral pH is maintained here, flexibility is less. So interaction between enzyme and substrate is less. Very slowly interaction takes place between enzyme and the substrate. Then the reaction is very slow. So the enzyme activity is very less. The enzyme activity is very less. So what I am telling here, if amino acids are active, if active site is active, enzyme is active. Right? So here this is one of the graph. So here there is an y-axis is there. This is the y-axis and this is the x-axis. On x-axis we are taken that pH. There is in 1 to 14, right? And enzyme activity. Here this is the enzyme activity. One normal enzyme is taken, right? The majority of the enzymes are active in between 6 to 8. The pH 6 to 8. Between pH 6 to 8, majority of the enzymes are active. So based on this graph, actually what kind of graph we are obtained? If we construct the graph, pH versus enzyme activity, the shape of the graph is bell shape. This is the bell shape curve. So we get the bell shape curve, right? So here majority of the enzymes are active at 6 to 8 pH, right? So here at acidic pH the enzyme is less active. Gradually at neutral pH the enzyme is more active. Gradually at basic pH the enzyme is again less active. So what I am telling here, if we construct the graph pH versus enzyme activity, we get the bell shape curve. That is about the graph pH versus enzyme activity, right? Coming to the next one, effect of temperature on enzyme activity. Effect of temperature on enzyme activity. So here also same case. Here there is an x-axis. We have taken here different temperatures. And y-axis. Here this one is the y-axis. On y-axis we have taken the enzyme activity. Right. So if we construct the graph between these two, or the temperature versus enzyme activity, if we construct the graph, we get here also bell-shaped curve. Here also we get the bell-shaped curve. So what I am telling here, there is an temperature and enzyme activity. We get the graph bell-shaped curve, right? So here also a group of enzymes are active at between 40 to 45 degrees centigrade. Majority of the enzymes are active at 40 to 45 degrees centigrade. At low temperature enzymes are inactive. At high temperature enzymes are inactive. The best example, when a person gets the fever, or any person suffering with the fever, that time the person is very weak. Okay, the person is unable to walk properly because inside the person body, okay, there is no production of energy. Why energy is not produced inside the patient? Because here whatever that enzymes are there at high temperature, the enzymes are not working properly. At cold conditions also, see, sometimes during the winter season also we feel very weak and very lethargic. Because here when the temperature is less, the enzymes are inactive. When the temperature is less, the enzymes are inactive, right? So what I am telling here, at low temperature, at high temperature, enzymes are inactive. Sir, why enzymes are inactive at high temperature and the low temperature? We already discussed the enzymes are having primary, secondary, tertiary structure. The tertiary structure is very important. At low temperature, there is no flexibility within amino acids, and at high temperature, the tertiary structure is destructed. Is there any structural changes that takes place, especially at high temperature? At low temperature, mostly there is no flexibility between the amino acids. There is a freezing state is there. So there is no movement of amino acids. The substrate is unable to bind with the enzyme. Then there is no reaction. So that's the reason why at low temperature, at the high temperature, enzymes are inactive. Whatever that average temperature is there, 40 to 45 degrees centigrade, at the temperature, enzymes are active. Right? So that is about effect of temperature on enzyme activity. Coming to the next topic, substrate concentration on enzyme activity. Remember here, enzyme molecules are constant. In a particular reaction, number of enzymes are constant, or enzyme molecules are constant. Then we are increasing here substrate concentration. For suppose here, number of enzymes are 10. Okay, the very first substrate molecule is 1, 2, 3, 4, 5, right? Continuously we are increasing the substrate concentration. So we are constructing the graph substrate concentration versus enzyme activity, right? So we get here, there is a steady state graph. See, this is the starting point. Up to one point, the enzyme activity is increased. If we increase the substrate concentration up to certain level, the enzyme activity is increased. After that, then we get there is a plain line. Or steady state line. Because I already said enzyme number is constant. We are increasing the substrate concentration. After certain limit, if we increase the substrate concentration, then there is no use, because here always enzyme concentration limits the enzyme activity. Here in this reaction. We provided all the favorable conditions. We are continuously increasing the substrate concentration. Enzyme concentration is here constant. Number of enzyme molecules are constant, right? So we get that steady state level. See the final point, this is called as Vmax. Vmax indicates here maximum velocity of reaction. Is called as Vmax. For suppose in this graph, the maximum velocity is the 10. See, Vmax by 2. Vmax by 2. This is the Vmax by 2. So the value is 5. Here Vmax by 2 is called as Km. Vmax by 2 is called as Km. For suppose this is the 10. Okay. Then here maybe this is the 5. Then we draw the line. The point is indicated here Km. The point is indicated here Km. See what I am telling here. The maximum velocity of reaction, this is called as Vmax. Vmax by 2, that is called as Km. Vmax by 2 is called as Km. So of maximum velocity, Vmax by 2 is nothing but of maximum velocity is nothing but Km. Km is also called Michaelis-Menten constant. Km is also called Michaelis-Menten constant. So here in this graph we have to remember 2 points. One is called as Vmax, maximum velocity of reaction, that is called as Vmax. Vmax by 2, that is called as Km. Vmax by 2 is called as Km. This point is exactly Km, right? Of maximal velocity or of maximum velocity, that is called as K. Vmax by 2 is nothing but of maximal velocity. The point is called as K, right? See, Km is also called Michaelis-Menten constant, right? That is about the substrate concentration. That is about the effect of enzyme activity by the substrate concentration. So here simple. If we increase the substrate concentration, okay, the enzyme activity levels up to certain point level. After that we get the plane or straight line. After that one we get the straight line, right? That is about the substrate concentration. Effect of substrate concentration on enzyme activity, right? That is about the substrate concentration. Next one, metal ion concentration. Here metal ion concentration. We already discussed a group of enzymes are there which are interact with that metal ions. Presence of enzymes, the enzymes are more active. We know that salivary amylase enzyme requires chloride ions. Salivary amylase enzyme requires chloride ions. In presence of chloride ions, salivary amylase enzyme is more active. So here also a group of enzymes are there for their enzyme activity. The enzymes for their enzyme activity or more activity, metal ions are required. So iron, here Fe plus 2, that is okay, that one is required for cytochrome oxidase enzyme. Cytochrome oxidase enzyme. And zinc, there is a zinc enzyme, Zn plus 2, carbonic anhydrase enzyme. And next one, magnesium, Mg plus 2, right? So hexokinase enzyme. So hexokinase enzyme, activated presence of magnesium ions. Carbonic anhydrase enzyme, activated presence of zinc ions. Cytochrome oxidase, activated presence of, more active presence of iron. So pyruvate kinase enzyme active, presence of potassium ions. Arginase enzyme active, more active presence of manganese. And nitrogenase enzyme, the enzyme involved in the nitrogen fixation process, right? So it requires molybdenum ions. And last one, glutathione peroxidase enzyme, that one is requires selenium. That one is requires selenium. So different types of enzymes requires different types of metal ions. Presence of these metal ions, these enzymes are very active or more active, right? So that is about the metal ion concentration. Metal ion concentration affects the enzyme activity, right? Okay. Coming to the enzyme concentration. So we are already discussed. More enzyme concentration is there, more rate of reaction takes place. In a reaction, if we increase the enzyme concentration, we are adding more number of enzymes. Okay, then the rate of reaction also increased. And coming to the effect of time. Okay, here always any reaction requires time. Either enzymatic reactions or non-enzymatic reactions. Okay, requires time. So effect of time in a particular reaction. More time is available for the completion of a particular reaction. More time is available. More rate of reaction will be takes place. So presence of enzymes, more time is available. More rate of reaction takes place. So effect of concentration. If we increase enzyme concentration, more rate of reaction takes place. If the enzymes are for that reaction, more time is available. More time is available. More rate of reaction takes place. So that is about factors affecting enzyme activity. That is about the factors affecting enzyme activity, right? So that is about that different types of factors which affects the enzyme activity. Remember, always enzymes requires favorable conditions. at favorable conditions. Enzymes are shows more activity. Enzymes are shows the more activity. For different enzymes, different activities are there. For different enzymes, different conditions are required. Different conditions are required.